All right, so we are already on live.
Okay, yeah, maybe we can get started then. I still don't see it live, but John said it's live, so that's good. Okay, yeah. Okay, yeah, welcome everyone. I couldn't be here last week, but now we're here. So welcome to the seminar, second lecture. It's good to see some people in person. I think sometimes we will have seminars in person also, and we'll figure out the infrastructure to both do live stream and in-person delivery at the same time. But today we're going to go with online delivery, but in-person attendance as well. But in future, we may have some seminars in person. If you want to deliver your seminar in person, for example, that's perfectly fine. We just need to ensure that everybody can see it. So you will need to be connected to Zoom as well. So I think what we will do today is Mohammed will give an example paper review. Hopefully this will serve as an example for you to deliver your talks. Also, you have a lot of examples, as Mohammed mentioned in the prior meeting. You have a lot of online examples, if you will, but this will be a live example of doing a paper review. And he's lucky because he's going to review one of the papers that he has written during his PhD, GateKeeper. So hopefully he'll put a critical eye on it and talk about the weaknesses as well. So even if you're reviewing your own papers, it's important to talk about the weaknesses of the work, because that's always both scientific and also it enables future work, right? Okay. So Mohammed, are you ready? Okay, maybe after Mohammed's, there's a second part of this meeting. After Mohammed finishes the example review, he will also lead a discussion on important issues in accelerating genomics. Is that correct, Mohammed? You're muted. Okay.
Yes, that's true. Thanks.
Okay. Okay, yeah, and I hope people will participate. Let's try to make it lively. I mean, due to COVID constraints, we have to be hybrid, regardless of wherever you are. Please participate in the discussion. And I think when you're speaking, it's always good to turn on your camera. But again, if you're uncomfortable, you don't have to do it. But it's a good suggestion, I think, so that everyone can participate, let's say in a nicer way. So the second part is going to be hopefully a good discussion led by Mohammed. Okay, Mohammed, go ahead, please.
Thank you so much, Onur. And I'd like to welcome everyone to the second lecture in Seminar Computer Architecture course. I really encourage everyone to just stop me and ask questions whenever you feel you have some concern to share or feedback. All right, so as Onur mentioned, this is the example for a paper presentation where we will discuss how to present, how to analyze a paper. So the paper for today called GateKeeper, a new hardware architecture for accelerating pre-alignment filtering in DNA short read mapping. So the first slide when we present a paper in this course should be the cover slide where we present the title, the author list, the venue, and the student name who's presenting the paper. You could show some affiliation, or you could, it's optional, the template basically, or the formatting, but feel free to show these important things. All right, so I'll start officially now, and maybe at some point I will give feedback on how to prepare your slides. All right, so the first thing, the executive summary. So the problem that this paper is trying to tackle is that genomic similarity measurement is a computational bottleneck. And examining the similarity of highly dissimilar genomic sequences consumes an overwhelming majority of modern read mapper execution time. And basically in genome analysis, it's very fundamental to compare genomic sequences together, aiming at finding, for example, the similarities or the differences. And it's a problematic in case of highly dissimilar genomic sequences, because those sequences normally don't lead to any genetic discovery because they are really different. So probably this is not the good comparison that we are aiming for, and we are looking only for those sequences that are closely to each other. And the problem is that we are using a computationally expensive algorithm to check the similarity. So we don't know in advance whether these are similar or highly similar or highly dissimilar even. And when we use very computational expensive algorithm to figure out that the two sequences are highly dissimilar, then we ignore these sequences and move on to the next sequences. That's why this is a waste of time, because we wasted a lot of computation for just ignoring these sequences at the end. And the goal in this work, as you can guess, is to develop a fast and effective filter that can detect these problematic sequence pairs, which we call them highly dissimilar from each other, and eliminate them without using computationally expensive alignment algorithm. Don't worry about the terms. I'm going to explain most of them during the background section. So the key observation we made in GateKeeper is that if two string or sequences, genomic sequences, differ by e edits, edits could be differences, then every pairwise match can be aligned in at most 2e shift. So you need to shift the sequence at most 2e times, e times to the right side and e other times to the left side. And then in this way, you can always measure the similarity correctly between these sequences if they have a number of differences that is equal or less than e, which is the threshold. And based on that observation, we make three key ideas to develop GateKeeper and solve this problem efficiently. The first one is to quickly find similar sequences using Hamming distance. And as you know from previous courses, that Hamming distance is just XOR of the pairwise characters. And if the goal of using Hamming distance is basically to find those are the similar sequences. But if they are not similar, if the differences between these two sequences exceeding a threshold, then we need to move on to the step 2, which is computing the shifted Hamming distance for the rest of sequence pairs. And basically, shifted Hamming distance is ANDing several masks together to identify these similar sequences. And to efficiently implement these two key ideas, we have to maintain a bit parallel operation in all of the algorithm that we develop in GateKeeper, such that we can nicely map the operation to SIMD instructions, for example, or FPGA, logic layer of the 3D stacked memory, or even in-memory accelerators such as Ambit. And the results or the benefits we gain from GateKeeper is really significant, such that providing a huge speed up of up to 130x compared to the previous state-of-the-art software solution. We also have comparison with the hardware accelerators and so on that I'm going to show through the evaluation. So that was for the first slide, which is executive summary. I really encourage everyone to have that slide in their talks. And now I'm moving to the background. And since this topic is from genome analysis, which we don't expect the audience to have a solid background in genome analysis. So basically, I'm going to provide a thorough background in detail about genome analysis, read mapping, and all the terms that I promised to explain them. All right. So what is genome analysis? And as you can see from the picture, there are multiple people that really have different phenotypes, traits, and so on. They might have disease. So out of curiosity, we would like to know.
I was supposed to come look at my exam.
Is there a question?
Your exam today.
About your email.
I think that is Jisung.
With me?
Yeah. You're part Jisung.
Yeah, sorry about that.
Yes. All right. So the genome analysis is basically to identify what caused these differences between people. So we'd like to know whether these genetic variations causing these phenotypes or disease or something else. And basically, to analyze these genomes, there are multiple benefits to do so. So the first one could be, as I mentioned, understanding genetic variation, or it could be predicting the presence and relative abundance of microbes in a sample. So think about going to Zurich Hauptbahnhof, just collect a sample from train door or train station, and you'd like to know whether there are some harmful viruses or bacteria exist in such samples. So the first step to do is genome analysis. So you sequence that sample, or you try to read the genetic material and then analyze it and figure out which virus exists in this sample. And this is what we call metagenomic profiling analysis. You could also use genome analysis for rapid surveillance of disease outbreak, as the time we are in currently for COVID-19. So you'd like to analyze sewage water or the air condition and try to figure out whether COVID-19 exists, or maybe the other bacteria that exist with the COVID-19, such as a lot of studies for human microbiome gut analysis and such. You could also use it for developing personalized medicine. Think about having historical genetic data from the hospital or previous patients, and then you got now a new patient admitted to the hospital. So giving the genetic material of the new patient, you could refer to the historical medical data and see that if they have these genetic variations and the previous patient were getting better because they got a certain medication, then probably the new patient will get also well and having good outcomes just if we give them the same medication. And that's what we call developing a personalized medicine. So it's really important to have genome analysis in all aspects of life. That's why we are enabling or trying to enable this to be a very cheap way to do genome analysis, cost-effective, energy efficient, and so on. And this is our goal, basically. You provide the genetic material, it could be blood, and then the output we expect it to be the full sequence of your DNA, for example. Unfortunately, we don't have such machine, as I explained in the first lecture. And with that, now genome analysis is very challenging. So this is a commercialized kit that you can get it online or from pharmacy. We are not doing advertisement for them, but this is how, if you would like to do genome analysis, for example, you take a swab, you send back the sample to the company, and they will do the analysis for you. And what they will do, basically, they have one of these machines where they pre-process the sample you sent in the lab, and then they put it in this machine and they try to generate data out of it. Unfortunately, the data they generated from these machines, that we call them sequencers, they don't have any information about their order or their location. So it's really a mess. These readings happening randomly for the entire reference genome. You don't know which piece coming from which place. And for that, you really need to sort them and find the right place for each of these pieces, such that you can get back the complete sequence of your DNA. So I like this analogy where you can think about it as solving the puzzle. So imagine that you have these pieces and you would like to connect them together. And for that, you need basically the reference picture so that you can follow it and compare each piece to the entire pieces of this reference picture, and then try to figure out what is the right place for that. And after you solve the puzzle, you may have some gaps, you may have some errors, exactly what we have in the genome analysis. So these sequencing machines normally produce some errors such that they affect the accuracy of your DNA sequence. All right, so let's come back to genome analysis. Now we have the reference genome already available. We spent 13 years to produce that reference genome, and it was almost ready in 2003, but it wasn't complete. And this year, they try to bridge the gaps and they produce nearly complete, or let's call it a complete reference genome. And the reference genome is normally in a FASTA format, which is 2-line based. The first line is the ID, or the unique identifier, and then the reference genome as one sequence split it over multiple lines. This is an example where you can download such reference genome. It's already freely open, available publicly through NIH website. And these are the pieces produced by the sequencer machine, which we call them reads. And normally it's in FASTQ format. So the reference genome was in FASTA format and the reads in FASTQ format. For the FASTQ format, it's basically 4 lines per read or per piece. And these 4 lines, the first one is the ID. it's exactly same as the reference genome. And then the second line is the read itself, the genomic sequence that we got from the sequencing machine. And then the third line is just a separator, just a plus sign. The fourth line is the quality score. So each character in this quality is basically the confidence of the sequencing machine when generate the corresponding character. So it's basically in ASCII format. So for each read, we have 4 lines. So you can think normally about the size of these files when we get from sequencing machine. This is, again, another example for the read set that you can get from real machines. So you can see the machine name here and the specs of the sequencing run, what's happening over there. You can change this accession number at the end of the link and you get another data and so on. Okay, so now we have the reference genome, we have the FASTQ file, and you'd like to specify the location of each read in the FASTQ file in comparison with the reference genome. So we would like to map them. That's why we call it a read mapping step. So we have the genetic material from the subject individual. So we sequence it, we got the read, and then we got the reference genome, and we would like to map these pieces to the reference genome. So we try to compare each piece with several locations in the reference genome until we find the optimal location or the best location. And we keep doing that for all pieces until we get a nice complete sequence of the subject individual. So when we do the performance analysis for this step, which we call it read mapping, and we compare it to the sequencing, for example, we find that the sequencing machine is doing really well. So it can perform or sequence a complete sequence in 1 hour, a complete human genome in 1 hour. But the analysis, you can do only 1 human genome within 32 CPU hours. That is in a 48-core processor. So you can see the gap is really far, is really widened between the sequencing machine and the performance of the analysis. Okay, what makes read mapping a bottleneck? So first, we need to learn how to map a read. And as I mentioned, we need both the reference genome and the read set. And since the reference genome is really huge, it can be in size 3.2 gig, and you cannot normally build a hash table, for example, or any kind of indexing for the entire reference genome, because it might be very large and then it will not be efficient. For that, we collect only few pieces, subsequences, very small subsequences. Each subsequence can be 20 in length, and then store these somewhere. Imagine you have some index structure where you store this piece and its location in the reference genome. Now we do the same from the read set, the FASTQ file. So we collect these pieces of same length from the genomic sequence, which is the second line. And then we use these pieces to query the pieces that we stored from the reference genome. If we find them, if the location of these pieces makes sense, for example, here we can see the order of the blue piece, then the green, then the red, then possibly this sequence over there is where we got our read from. And then we do the comparison between the read and this target, a subsequence from the reference genome. We do what we call sequence alignment, which is normally dynamic programming algorithm, such as edit distance calculation or alignment score and so on. This is what we do after we detect the location in the reference genome. We try to investigate further and check base by base comparison. And when I mention edit distance, what I mean is that the minimum number of edits, which can be insertion, deletion, or substitution, needed to make the read sequence exactly matches the reference segment. So think about, for example, organization and operation. So you'd like to map them together, assuming there are some genetic variation that makes them different. Then you would like to know where are these differences and what type of differences we have between the organization and operation, such that we can make them similar. So you can see there are different ways to do the operation. For example, here deleting 2 characters, here inserting 5 characters, and so on. You could do it in another way. And here is another example where we have 3 alternative ways to map organization to translation, for example. Now, which one to pick? That's another question. We need to follow what we call alignment score. Which one is giving the highest alignment score? Then this is the optimal sequence alignment solution. Okay, hopefully that was a brief background on how to map the read to the reference genome. Now let's go back to our question. What makes read mapping a bottleneck? The first one is that we have a tsunami of sequencing data. And that was clear from the performance of the sequencing machine in comparison with the genome analysis or the read mapping performance. And for that, the sequencing machine keeps generating a huge amount of data every second, but the analysis is done really at the bottleneck, such that we cannot cope with the scale of the genome sequencing. The other thing is that we are using specialized machine for sequencing, but when it comes to the analysis, we are using normally general purpose computers to do the analysis, and which are not sophisticated to do the genome analysis, for example. And that is fundamentally the reason why it's really slow. And when we move next to the CPU and we do the analysis, we see a huge amount of data moved from one part of the compute stack or the CPU parts to the another, especially when we are talking about a huge amount of data moved from the sequencing machine to the storage unit, to the main memory, then to the caches, to the CPU, just to detect that these sequences that we moved all the way were highly dissimilar. And then we just reject them. So we waste a long journey just for detect these sequences. So it will be more efficient if we can detect these sequences, for example, at the storage level or at this inside the sequencing machine itself. So the data movement is always expensive to perform. So single memory request consumes about at least 160x more energy compared to performing an addition operation at the CPU side. And that's why data movement dominates performance and it's a major energy bottleneck, which is at least 40% of the energy just spent on data movement without performing any computation. Now, when we do analysis for the read mapping software, which we choose here as Minimap2, which is state-of-the-art and widely used by even the company ONT, which is producing the sequencing machine, we find that more than 60% of the execution time is just spent on the dynamic programming part. That is including this sequence alignment and chaining, which used to do some filtering or some alignment. Let's call it sparse dynamic programming alignment. So there are 60% of the time just spent on sequence alignment. And as I mentioned, there is a large portion of this time also spent on finding the highly dissimilar sequences. Now, what makes this alignment is computationally costly? First, it uses dynamic programming algorithm, which is well known for quadratic time performance. And we cannot say this is a bad thing. This is actually a very good thing. Why is that? Because first, think about this example. We have Netherlands and Switzerland, and we would like to check the similarity or calculate the edit distance between these two words. So how you would like to do it is compare Netherlands with S, then with SW, then with SWI, and so on until you figure out the entire comparison with the whole word, which is Switzerland. Now, apparently you can see a lot of repeated operations. So you already compare with the S here, and again, in the next step, you are going to compare with the S, and then with the W, and then with the SW together. So dynamic programming sounds like a good solution for this case, because you don't need to repeat all these operations all the way until you figure out the edit distance, but rather you store it in a table. And whenever you need to compare with the S again in the next step, you just go back to that cell in the table and reuse that value. So dynamic programming algorithm is not bad, but actually the way we use it, it's bad. So the second problem is that there are a lot of data dependencies that limit the computation parallelism. So you can see here in the example, since we are filling this table, so there is a strict order on the way we compute this table. For example, we need to compute the first cell, then the second cell in a row, and then the third row, the third cell, and so on until we compute the entire row. And then we move to the next row and so on. Or you could do it column-wise. So you finish the first column, then you move to the first cell in the second column, then to the second cell in the second column, and so on. You could do it anti-diagonal, for example, and so on. But you cannot calculate any given column without calculating the previous or the neighbor column in advance. And that limits the way you can parallelize this computation. Now for the third point is that sometimes, especially when you do global sequence alignment, which is one type of doing sequence alignment, is that you need to compute the entire matrix, the entire dynamic programming table, such that you figure out the optimal number of edits, which is in this case, it's the very last cell in the dynamic programming table, which is 5 here. So the optimal number of edits between Switzerland and Netherlands is 5 edits. And for that, if this is exceeding a threshold, you already compute the entire thing until you figure out that these differences already exceed the threshold. And now this is for the new challenge or the new problem that makes read mapping is a bottleneck, that 98% of the times that you compare genomic sequences together, we find these sequences to be highly dissimilar. So we use dynamic programming algorithm to figure out the match and mismatch between the two sequences, but 98%, which is vast majority of these sequences, are highly dissimilar. Then this is again wasted time. So we only need optimally to use dynamic programming for 2% of the times we compare genomic sequences together. All right, so in conclusion, we need intelligent algorithm and intelligent architectures that handle data well. So the algorithm should be aware about the architecture, and the architecture should be supported by the algorithm operation. And we need to handle data well, because you can see there's a huge data movement from one component to another. And without knowing what type of data we are handling, or what is the problem, then we cannot design an algorithm that is efficient even for CPUs. So there is a detailed analysis of tackling this bottleneck in a paper that we published recently. So I recommend everyone to go through this paper if you are interested in this topic. And now let's continue with GateKeeper. So our goal is to minimize alignment time. So we find out sequence alignment is very expensive, and our goal is to accelerate read mapping by reducing the need for dynamic programming algorithms. So we want to find not the alternative, but something that comes in action just right before using the dynamic programming. And we will use that magic or that thing that we will design to filter most of the things and keep the correct ones only for the dynamic programming algorithm to operate on them. As I said, feel free to stop me whenever you have questions. And I think someone is watching the YouTube comments, if we got any comments from the YouTube audience. Okay, now let's move to the novelty of GateKeeper paper and the key approach and the ideas. Again, let's recap with the genomic strings. There are two types. There are similar strings that they have matches and they have mismatches less than a threshold. And we have dissimilar strings that the number of differences between them exceed the threshold. So as we said, for the dissimilar strings, we ignore them basically because the number of differences exceed the threshold and they don't have any interesting meaning for genomics. For the similar strings, we want those guys. So we want to keep them. We don't want to remove them or filter them out. And that's why these dissimilar strings are expensive, because whatever computation you use to calculate this dissimilarity at the end will be wasted because we are going to ignore them. And our basic idea is simply to filter these dissimilar sequences, but there are 3 metrics used to the filtering. So the first one, we want to filter out most of the incorrect mapping. It's okay to not filter them all, because otherwise the filter will be very efficient and might not be filtered, might be a sequence alignment if it is really efficient this much. And the second property that we want to maintain is to preserve all the correct mappings. As I mentioned, there are similar sequences that we want to keep, such that we figure out the genetic differences and matches. And we want basically to do this way quicker than the sequence alignment itself. We don't want the bottleneck to be shifted from sequence alignment to filtering, basically. Or we don't want to make the read mapper slower just because we add a newer step. And our proposed solution, or the architecture of GateKeeper, we implemented an FPGA, and that was the first FPGA-based pre-alignment filter in that time when we published the paper. And now I'm going through the mechanism or how GateKeeper works. Okay, so as I mentioned, I'm going to explain the observation I mentioned in the first slide. When we have two strings and the number of differences equal or less than E, which is our threshold, then we can match these two sequences correctly using only at most 2 E shifts. And the key ideas is first to quickly find similar sequences using Hamming distance, and then compute the shifted Hamming distance, and then use bit parallel operation to operate the GateKeeper algorithm. Okay, so let's explain the first observation. Now imagine you have two words, Istanbul and Istanbul. You would like to compare them and see how many differences between these two words. They are exactly matching each other, right? So if you do Hamming distance or just XOR operation between these characters, you get 8 matches and 0 mismatch. Now we would like to impose ourselves a genetic variation or a difference. Let's delete, for example, the letter A, and we want to do the comparison again. Now, since we do the deletion, this is a new word without the A, and when we do the comparison, we find that there are only 3 matches and 5 mismatches. Just we did single modification and we got 5 mismatches. That caused by the deletion. That's why Hamming distance won't work here. So what we will do, we will use a shifted version of the word Istanbul after removing the A. So now this is a shifted version of it. We shifted to the right by 1 step, and I'm going to explain why we shifted to the right, not to the left later on. But now when we do the comparison, with the first version, we find there are 3 matches, and we do the comparison with the second version, pairwise comparison, character to character, and then we find there are 4 another matches. And now when we convert into bitwise XOR, we find out when we do the AND operation between these 2 XORed values, then we got this pattern. So we got 3 zeros, 1, 4 zeros. And those at the same location, they indicate or represents the corresponding operation that we did. So you can see at the 1, we deleted the A at the exact location here. And these zeros represent the exact match. And this is the mismatch. And when we quantify the number of zeros, we find that there are 7 matches and a single mismatch. And this is now the correct comparison, not as before, where we have 5 mismatches. So we will use the same analogy to solve the filtering issue that we have. And for that, we did. Now let's take a real example and let's see how we perform this shifting on real example. So you have a query and the reference sequence or a subsequence of the reference genome. The reference genome, again, is a huge, like 3.2 billion characters. So we got subsequence of it, and we try to do the optimal comparison with the read. Now what we build, these are the 2 E plus 1 bet vectors. What is E again is our threshold. How many errors you want to tolerate. So here we specify it as 3, then we have 7 bit vectors. The first one is XOR, which is the original version of the comparison between the query and the reference genome. And the second 3 bit vectors are the shifted version of the query and then do XOR operation. So we shift the query by 1 step to the right, and then we compare it to the reference. And then we got the second mask, which we call it 1 deletion mask. The second or the third mask, which we call it 2 deletion mask, is generated after shifting the query now by 2 steps, not 1 step, shifting the query by 2 steps to the right, and then do the XOR with the reference. This is how we got the third bit vector. We continue until we finish all the deletion mask. And now when it comes to insertion mask, for example, 1 insertion mask, we shift the query to the left, not to the right. And then we do the XOR operation with the reference. And the 2 insertion mask is exactly the same thing, but we shift by 2 steps to the left and then we do the comparison with the reference using XOR, and we calculate this bit vector. Now, maybe someone of you will ask me why we did several or a set of masks as a result of shifting to the right and the other set shifting to the left. Why not always shifting to right, for example, or shifting to left? So, because in the previous example, we did the difference or the deletion ourselves, so we know where is it, and we know the type of that modification we did. But here, in real example, we don't know this information in advance. We don't know whether the genetic variation is because of a deletion or insertion or multiple of insertions, or even a combination of these insertion, deletion, substitution together. That's why we have to take all possible ways, assuming that we have at most 3 consecutive edits, because we set our threshold to be this, but we can set it to 10, for example. But then we need to generate 21 bit vector. Okay, I hope it's clear how we generate all these bit vectors. If you have any question, just feel free to ask directly. You can enable your mic and just speak up. Okay, so now I would like to highlight something. These consecutive zeros. So you could think, what are these zeros? You can see a lot of random zeros, short segments of zeros everywhere, but these, very interesting because they are long. So these represent the exact matches between the query and the reference subsequence. And our goal is just to collect them. We would like, since these represent the exact match, so if we collect them, we quantify how many zeros in these segments, the green highlighted segments, then that gives you an indication about the differences, because the total length of the query minus the total number of these consecutive zeros will give you the number of differences. So our goal in this work is to collect these segments. And how we do it, as I mentioned in the first slide, we are going to do AND operation between all these masks. And what we got is this result. So you can see we got a large number of zeros and we got 2 ones. These ones, again, represent the number of differences or the optimal number of differences between the query and the reference. Now, you can think about these 2 ones, for example. So you can see here there are 2 consecutive ones splitting these 2 green segments. And if you investigate it further, you will see that the query is really different from the reference at this location. But when we do the AND operation, we did not get anyone in the corresponding location, which is here. I hope you can see my cursor moving. So that is an inefficient way of implementing the algorithm, because the AND will always dominate whenever we have 0. So the 0 will dominate any one in the column. For example, any given 0 here in this column will always dominate, and we'll never get the 1 in the final result. For that, we did a heuristic that we replace any pattern that is 1, 0, 1, we replace it with 3 ones, or 1, 0, 0, 1, which is 2 zeros in the middle of 2 ones or surrounded by 2 ones, we replace them by 4 ones. Why we did that? So let's see the next figure. You can compare now the bit vectors with the previously generated bit vectors. You can see the number of random zeros here are everywhere, but here it's a bit cleaner, a version of these masks. How is that happening? As I mentioned, we replace any single 0 or 2 zeros by 1s. Why is that? Because we want to allow anyone to appear in the final bit vector. So we want to cancel the domination of zeros. And whenever we do the AND, then we will get a 1 at the end if there was a real 1, not the one caused by our own modification. And when we did the AND again now, we find that this a bit good solution, but is not solving the problem completely. So you can see still we get single 1 in the final result, but not 2 1s, which is the optimal solution. That is fine because we are not looking for optimality here as long as we have correctness. So we want to build a filter, but not the sequence alignment. It's not the final step. It's just a filter to remove most of the wrong data for the dynamic programming. And the dynamic programming will be responsible for the optimality part. This is our goal. All right, so the third key idea is to use only bit parallel operation that nicely map to most of the hardware acceleration platform. And this was nicely shown by the algorithm. So you can see most of the operation that we use in our algorithm were basically bitwise operation, shift, and, or, XOR, and so on. And for that, we choose FPGA platform, which will nicely be the best solution for bitwise operation. Now, we talk about the problem of dependencies between the sequence alignment in the previous slides. Now, how about now? How about the bit vector or the mask that we generated? Do we have any data dependencies? So remember, when you calculate any cell here, you want to calculate the previous row or the previous column. But here, in our case, so we do shifting to the entire sequence. For example, this one, we shift it to the right by one step, and then we do the comparison. So there is not really much of data dependency. There is none at all, actually, any data dependencies. You can solve any of the cells before the rest of the entire table. So for example, C, you can shift it, if it is here in this corresponding location, you can shift it by one step and compare it with G directly and compute this cell. And that is helping us a lot when we do the data parallelism or the computation parallelism. So you can see you can compute the entire thing independent of any other cells in the dynamic programming table or in our mask calculation. And as I mentioned, for that, you can nicely implement the algorithm for FPGA, GPUs, SIMD instruction, and so on. Now I'm going through the hardware architecture. If you have questions, I would be happy to take them now before moving on. I hope the algorithm was clear. Yeah, I see one student in the classroom.
So I have one question regarding the amending part with the 101 and the one. So why I didn't catch why we do that. Could you maybe repeat that? And after that, could you maybe repeat how the algorithm with the less data dependencies work?
Mm-hmm. That's a very nice question. Two questions, basically. What's your name?
Daniel.
Okay, Daniel. Yeah, good one. This is the problem of having... So we cannot see your name from the Zoom call. Yeah. So let's move to the previous slide. Yes, here. So now, I hope this problem, the one I highlighted with the red box, was clear?
No, actually not really.
Okay. All right. So when we do the comparison between the query and the reference, there were some differences. Let me highlight them maybe. Okay. So there were differences over exactly, I would say, so probably it's here around these two characters or shifted a bit to some other two characters. And these differences, I think it's GGA. Yeah, probably it's around here. When you shift this sequence, the above one, the query, to the right by one step, you'll see it clear that there are two differences between them. And these two differences appearing here clearly, right? Because the second mask, is where we compare the shifted version. So after we shifting the first one, which is the above here, the sequence, the above sequence, we shifted by one step to the right, and then we do the comparison. So if there is any difference, it will appear directly on the second mask, not the first one, because the first one represents a comparison without shifting. So I think there was a deletion here when you compare it to the first sequence. Yes, I think there is a character deleted, which is the C. So you can see here, there are two, maybe you can remove this. Yeah, there are two C's here, but there is single C here. So there is a C that was deleted from the query. That's why the matches, we got them in the second mask and not continuing in the first mask. So imagine if the query and the reference were exactly the same, then the first mask will be entirely complete of segments of zero. There is no ones at all. because it's exactly matching, right? And the first mask is just XOR operation. Now, because we have a deleted character here as a highlight, I don't know where is the cursor. Cannot see the mouse. Yeah, probably is here. No, it's here. So this character, the C, was deleted from here because this change from the segment above, the green segment, to the second segment. And that's why we got a green segment in the second mask. Now back to your question, we have here two differences. And yeah, I think I cannot see the cursor. Yeah, this is the problem when I use the annotation in Zoom. Let me delete them. Yeah. So back to these two substitution errors, I assume to get them here, which is the case. So I got 2 ones. But when I do the AND operation, you do not get them here in the result. You can see it's just full of zeros, right? And that was causing some error in the accuracy of the filter. Then the false positive, or the number of sequences passing your filter, not unnecessarily, will be really high. So most of the sequences that are dissimilar from each other will pass your filter. But our goal is to reject them. So if you allow for underestimating the errors, because here between the query and the reference, we have, I think, 4 differences. We have 2 here. Let me get back to the mouse. Yeah, I hope you can see the cursor now. We have 2 errors here between the sequences. We have 1 here was deletion, and we have substitution here. It's clear, you can see it between the G and the T around here. Can you see the mouse moving?
Yes.
The cursor, yes. Then we have 4 errors between the query and the reference. And our threshold was 3 when we calculate these masks. So if you use a threshold of 3 and you have a sequence pair with 4 errors, and you are estimating the errors to not be 4, but rather 2, then this sequence pair will pass your filter. And that is wrong. So we try to address this issue by converting every single 0 to a 1, because it's okay to have 1 rather than 0, because the 0 will cancel the 1s. Right? So we don't want.
Can you maybe repeat that? I think that was too fast, too much 0 and 1.
Yeah, so the 0 will dominate and cancel the 1s. Right?
Yes.
This is clear.
Yeah, that's clear.
So the problem apparently is with the zeros overlapping with the errors, which is 1. So we want to cancel the 0 such that when we do AND operation, we will not cancel the 1. We will not underestimate the number of errors. So we want to keep the zeros away from the ones that we would like to calculate. So here there are 2 ones. We want to calculate them, but we don't want any zeros overlapping with these 2 ones. So one of the heuristics is to replace these zeros with 1s, such that when we do AND operation, in the results, we will get the 1 if they are there, but we will not cancel them. We will not have zeros instead of 1s if there are genetic variation or if they are edits. I hope this was clear. If it is not clear,
no, I think it is clear, but doesn't this heuristic, you said it's a heuristic, doesn't this increase the chance that we throw away good ones as well? Or, so, well, what's the proof for us not throwing away good ones that we actually want to have?
Yes, that's a very good question. So there is a guarantee that this never rejects a sequence that is correct. Why is that? Because this is not going to replace the long segments of zeros. So think about the green highlighted segments, right? Can you think of a way that we will change these zeros to 1s? Unless our threshold or the pattern that we are looking for to replace is really this much long. Think about 1, then 10 zeros, then 1. If we replace this much of zeros by 1s, then we are going to overestimate the number of differences. But since the correct answer always is involved long segments of consecutive zeros, such as this case of this 1, the green segment here, then the second one over there, then the third segment over there, then the fourth segment over there. And all these segments have more than 2 zeros as part of the solution. So we are not replacing these. And this is a guarantee on not having false negative at all, or incorrect way of filtering the algorithm. Unless we increase our threshold for the pattern, that we replace, for example, every 3 zeros or 4 zeros by 1s, then we are going to hide some of the zeros that are part of the solution. And I told you these segments, the green segments, are indicating the exact matches between the query and the reference. For example, if, let me get back to the annotation. So this entire segment represents that this exact portion already exists in the reference as it is, without any modification. So you can see starting from here, no, from here. After the C, all the way to the T is exactly the same, or even to the 3 A's or 4 A's here. Exactly the same exists between the two sequences. And for that, we are maintaining this entire sequence as it is. And since it's full of zeros, it will always dominate. It will always cancel whatever ones you have here. And then we will have it in the final answer, and then we can quantify the number of zeros here correctly. Same thing with this sequence. Is full of zeros. We will never replace it. So whatever we do replacement here in this area, that green segment will always dominate, and you will have full segment of 0 over there, full segment of zeros over there, and so on here. And then the problem, or not the problem, the good thing that we are looking for will be in between these segments, which is, let me highlight it in red. It will be this region, and that region. Whatever we will have here, 1 or zeros, already indicating that these are modifications or genetic variation. Why is that? Because if they were not, then we will get a full segment of zeros instead of 2 segments of zeros, where we have in between them some differences. If these differences do not exist in the first place, then the 2 segments of consecutive zeros will be 1 long segment of 0 rather than 2 pieces of zeros.
Yeah, so I think you got a little bit clearer, so thank you. But could you maybe now explain the dynamic programming part again?
Yes, sure. All right, that was great. Thank you so much for this very nice question, basically. I hope it's clear now for everyone. I'll be happy to explain how it works after the class as well. But let's move to the second question. So for the dynamic programming, you cannot move to next row without calculating the first row, right? So you have to calculate the first row first, and then the second row. Why is that? You can refer to any dynamic programming equation, for example, Needleman-Wunsch or edit distance or Smith-Waterman, and any of these alignment algorithms. In the equation, you will see that when you want to calculate this cell, you always go to, let me annotate. I like the annotation in Zoom. So the first, no, this is red. Let's make it green, maybe. If you want to calculate this cell over here, we want to calculate this first, and this, and that one. So 3 cells to calculate 1 cell. Why is that? If you check the equation, you will see that you are accessing these cells in advance and check their values and add or subtract some value that we call it penalty. And then you take the maximum to calculate this cell. So that was a requirement for the dynamic programming part. Then you cannot calculate this cell over here, let me highlight it, without calculating the other 3 cells. That's why we cannot move from one row to another unless we complete the entire row here, and then we can calculate this one. Or we calculate this column and then that column. I hope this part is clear for now.
Yes.
Okay, now for the GateKeeper, let me move to the cursor mode. Yes, for the GateKeeper, we are shifting the sequence. Now let's get back to annotation. We shift this sequence to the right and then we do the comparison with this guy, right? So now it's okay to shift one of the character individually. For example, the C, we shifted alone one step to the right, and then we do the comparison with the T. So we can compute this cell. Now we shift the C one step to the right, and we do the comparison with the A. We shift the C to one step to the right, and we compare it with T. Then we calculate this one, right? I can do that. Still, the algorithm is correct. Now think about the T as well. So I don't know the cursor writes then. I cannot see it. Yes, now the T, the same thing. The T, the corresponding location is here in this cell. Now I can shift it to the right and then compare it with the T. I'll calculate this. Shift the T to the right, you can calculate the next cell and so on. So this is exactly the same. If I'm shifting the entire sequence by one step and then compare it to the this sequence, I will calculate an entire diagonal. And now when I shift the sequence, now you can see it. When I shift the sequence by two steps and then do the comparison, I will calculate the next diagonal, the entire one. When I shift the entire sequence, this one, by three steps and do the comparison, I will calculate the entire diagonal. Anyway, you do it this way, you shift the entire sequence or you shift character individually, you cannot see any dependencies between the cells. You can shift the C alone, compare it with the entire second sequence, cell by cell, or you can shift the third character, which is T, and you compare it with the first sequence and you calculate another cell and so on. That does not require you to calculate any previous cell in the table. You can see it clearly, maybe if you go back to the example. I will leave this maybe as a homework since we are a bit out of time. You can really do this yourself as a homework. You can go back to this example, take maybe the first 10 characters of the query and first 10 characters of the reference, and then try to do these calculations to calculate the masks again. And you can see it clearly that there's no dependencies at all. You don't need to calculate any cells in advance before calculating the current cell. I hope this was clear. Maybe it's a bit of more calculation. You can have an Excel sheet. Maybe I can share an Excel sheet with you where I calculate this example myself to verify the correctness, and then you can play with that. Or you could also play with the code. So the code is publicly available on CPU and FPGA. You can play with that and print this the entire masks and play with them. Maybe you can have another efficient, more efficient tool that you can publish and so on. This is how research started basically in our group. And by the way, some of the students present in the seminar course, we hired them at the end of the course and they continue doing research somehow in similar topics that they present in the class. So if you're interested in this topic, we would be happy to talk more about research ideas. I hope I answered the second question, Daniel.
Yes, thank you.
All right, great. Thanks a lot. Now let's move to the hardware architecture. Let me hide the annotation bar.
So Mohammed, there are some questions on Zoom also.
Oh, okay.
Look at them.
Thanks, Onur. Yeah, for Steve, how big is E chosen in practice? Okay, that's a good question. Basically, it depends on the sequencing technology we have. If it is producing short sequences, normally up to 5% of the sequence length. So if the sequencing machine produces segments of length 100, then E normally is 5 because the length is 100, which is up to 5%. For long sequences like Oxford Nanopore sequencing machine or PacBio, these are the two prominent companies in the market, depends on, again, the technology. So if it is ONT, Oxford Nanopore, they produce sequences with high rate of errors. Then we need to accommodate these errors plus the genetic variation. So if the error ratio is like 10% of the sequence length, then our threshold should be 15%, for example, because we need to accommodate the errors plus genetic variation. If it is PacBio, especially the hi-fi reads, it's way more accurate than ONT, for example, but the sequence length is moderate, like 10,000 instead of million. then we can pick a threshold of 5%, 6% around that. Let's say from 5% to 10%. This threshold can drop even to 2% and so on. It really depends on what you are looking for. If you are looking for genetic variation of type substitution only, then you could want to lower that threshold. Or you could, for example, generate only Hamming mask rather than shifted Hamming masks. Why is that? Because Hamming mask can nicely figure out where are these substitutions, but it fails when we have insertion or deletion. That's why we have several shifted version of the original mask to cover insertion and deletion. Is E the maximum of the longest possible DNA sequence that we want to find? Yeah, I don't know what do you mean by the question, but E is relevant to number of errors that we are trying to find rather than the sequence length. So the number of errors cannot be as long as the sequence, but normally it's portion of the read caused by some genetic variation or mutation or whatever the reason why we have these differences, but normally just small portion of the read covered by errors. Now the next question, I like this question basically. If we do the diagonal order, cannot we increasingly start to use more processes? So diagonally, you cannot do it, because in diagonal, you need to calculate one of the three cells after calculating the current cell. So you can do it anti-diagonal basically, or column-wise or row-wise. There are some research showing that you can divide the table into subgrid or submatrices of the table. So you can divide it over a group of cells together, and you calculate them, then you move to the next group and calculating them in parallel and so on. But you cannot do it diagonal. Must be anti-diagonal. You cannot do it in parallel, as I mentioned, but what you can do is use systolic array, for example, to calculate the entire row, but you cannot calculate the next row before the first row. It's a well-known method to use systolic array, which can feed the answer from one cell to the next cell and so on. And then you can pipeline the calculation for these rows or column. This is one way to accelerate it in FPGA. We can only use one process at the beginning and then use two processes for the child cells and so on. Yes, that is, if I understand it correctly, this is part of pipelining. So you could do pipelining. You can calculate the cell of the next problem while you are calculating the cell of the current problem and so on, where you pipeline multiple problems together, for example. How do we know that the two segments are aligned at the start of the sequence? We don't know that. We have no information at all. What are the type of errors, number of errors, and so on? We don't know that. That's why we really need to cover all possible scenarios. That's why we have 3 masks shifted to the right, another 3 masks shifted to the left, plus the Hamming mask. The Hamming mask always good to find substitutions. And in this way, all set together, the shifted to the right, shifted to the left, and the Hamming mask, they can find any errors correctly if they are less than the threshold or above the threshold. But remember, our goal is not to find them accurately or to find their type, because this is the goal of the sequence alignment, but not the filter. The filter only would like to know if the number of errors exceed the threshold or below a threshold. It's fine if we estimate the errors to be 2, while they are in fact 3, if the threshold is 3. So it's okay to underestimate them, as long as they are less than a threshold, that's always fine. It's okay to say they are 1 error or 2 error or 3 errors, as long as this number is always less than the threshold. Then we will never overestimate anything. Same thing, it's okay to say that there are 4 errors or 5 or 6 or 7 if the threshold is 3. Something above the threshold, it's okay to increase it by 1 or 2, that's fine. As long as the actual value or the correct value was already above the threshold. I hope I answered most of the question. Yes, if you meant the anti-diagonal, then yes, exactly. This is how we do it. It should be anti-diagonal, but again, you cannot calculate any anti-diagonal in advance before the preceding one. Otherwise, you need to pipeline the design. The editing distance is still calculated. Yes, exactly. After the filter, if the sequence pair has differences less than the threshold, then this is very interesting sequence pair, and we would like to know the type and the number of errors. That's why we moved to the dynamic programming and we calculated for these sequences. And this is not waste of resource because we are calculating for something correct. So we really want the result of the dynamic programming. We're not going to delete it, for example. For our GateKeeper paper, we didn't implement the sequence alignment on FPGA, and we have a nice argument for that. And we show it also with the later work. We have a lot of works to accelerate sequence alignment. We show that to do sequence alignment on FPGA, it's very challenging because either you are going to compromise the accuracy or some of the features and so on. And this is proven in the literature. You can nicely keep the sequence alignment to be done on CPU side because we do the dynamic programming only for 2% of the load, and 98% of the workload normally done by the filter. So we filter most of the incorrect things, and only we operate sequence alignment on the correct things. But again, that depends on the data, depends on the algorithm. If the read set coming, for example, from different species than the reference genome, then we expect the dissimilarity to be way higher and the ratio is higher. Then in this case, it might be more beneficial to have the sequence alignment, for example, on the FPGA, if the target two species are similar to each other, or the number or the accuracy of the sequencing machine is really high. Then the read and the reference are always similar, for example. Yeah, thank you so much for all these questions. If no more question, I can move to the hardware. Let's cover it quickly. So we have three main steps. First, generating these masks, and the next step is to do the change, the amendment for the single zero and two zeros into ones, and then do the AND operation. For the first one, generating the mask is a nice problem for FPGA. You can quickly do the shifting and the XOR operation. There's no problem with that. And this is how much resources you need to implement the first step. For the next step, where you replace single zero or two zeros, you want to do it in parallel. And what we use is lookup table. So we use 5 input lookup table, each lookup table operating on a single bit. So the input is 5 bits, but the output only single bit. You can see it clearly with the arrow. If you follow the arrow, maybe I can highlight it here. Yes, you can see it here, for example, this zero. That lookup table over here is responsible to calculate that zero. And why we need 5 input lookup table? So we are operating on all these 5 bits. The answer is, take this case, 1001, and don't care, and also this case. So to correctly calculate, this is the challenging case. The other case, 101, is not challenging. You can easily amend that. Probably you don't need 5 input lookup table. Probably you can handle it with 3 input lookup table, for example. But for this case, we require 5 input. And for that, we set the lookup table to be of 5 input 1. And why is that again? Because we need two bits to the right side and 2 bits to the left side of the target bit to know if this is 1001 or 1301, for example. If it is 1301, we shouldn't replace that. And that's why we need to check 2 zeros to the right and 2 zeros to the left. Because think about the case when we have 1, and we have, for example, 4 input lookup table, then you'll be in trouble. Because you cannot see the full pattern. You cannot see whether it's 1 3 zeros or 1 2 zeros, then 1. You can think about it later on. If you have a paper, you can write this example and see how it works. And this, why I want to highlight, although this is a very small thing in FPGA design, but I want to highlight it or emphasize it because this is very important when you pick the hardware. So it's really important to know what hardware you are targeting. If you are targeting FPGA board, although it has a lot of lookup table, but for the efficiency, you can pick the board that support at least 5 input lookup table. So there are boards with 7 input lookup table, with 4 input lookup table, and so on. We don't want to use 2 lookup table to replace this 5 input single lookup table. So for that, we choose Vertex 7 that has exactly 5 input lookup table. It can be configured to be 6 input lookup table. But because of that choice, then we do optimal resource utilization to the FPGA board. And that's why it's really important to know your algorithm and know your hardware as well that you are going to target. For ending the mask, this is how much resources we need. It's not really challenging. You can do the ending quickly in FPGAs. And this is the FPGA we are targeting. They have a huge amount of configurable logic blocks that are highlighted in blue. The rest is just to facilitate the communication between these blocks. This is again about the architecture inside the configurable logic block. This is single CLB, and you can see that has 5 input lookup table in each of these inside the slice. So this is why we choose this hardware platform. If you have any question, if not, I can move to the methodology. So this is the FPGA board that we targeted. It costs $5,000, which is not cheap, especially if we are comparing to the CPU, where we implement most of the bioinformatics algorithms. And we use this to do the communication, and these are the data set, which the real one is publicly available. For the simulated data, we already provide all command lines to regenerate or reproduce the data and test it yourself. And when we do the baseline comparison, we compare with the adjacency filter and shifted Hamming distance. Adjacency filter is slow and it accepts a large number of false positive, but shifted Hamming distance is more accurate, but it's limited with the length and it's slow because it's implemented for CPUs. In theory, GateKeeper is able to handle 140 problems in parallel. So if you have 140 sequence pairs, can do the filtering in parallel together. But unfortunately, we are limited by the data movement bottleneck from the DRAM to the FPGA through the PCI Express link, because the framework we used was limited. But these days, the framework to communicate data from the CPU to the FPGA way are really much better than what we used in 2014. So when it comes to reality, when you have actual implementation, not simulation, so we already connected with the PCI Express, and this is how much parallelism we can achieve to saturate the DRAM bandwidth or the data movement from the CPU to the FPGA. We could handle up to 8 and 16 problems, depends on the sequence length. If it is 300 in length, then we can handle only 8 problems in parallel. If it is 100 long sequences, then we can handle 16 problems in parallel. And you can see the utilization is really low for these 16 problems together. And if it is, it depends on edit distance as well. You can see the larger the edit distance, the larger the utilization, because the number of masks that we generate is 2e plus 1. If the edit distance is 5, then we are generating 11 masks for each problem. Then we are occupying more resources. This is about the throughput of the communication we have. And this is how much utilized from the FPGA chip if the edit distance is 15 and the read length is 300. And this is the key results of GateKeeper. So we, in total, or in summary, we achieved, move this bar. Yeah, we achieved 90x to 130x faster than SSD and adjacency filter, the state-of-the-art filters in that time, 2014. And when it comes to the accuracy, there is no false negative at all. What we have is false positive. That is also okay. What we mean by false positive is that when a sequence has more differences than the threshold. So sometimes we accept these sequences and we send them to the dynamic programming. But that is fine as long as this ratio of accepted sequences is as low as possible. So if we are sending like 100 or 1,000 sequences that they are different to the dynamic programming and the total workload is 1 million, then the benefit is still huge. especially if we are using a computationally inexpensive algorithm to do that job. And in total, when we integrate the GateKeeper to Mr. Fast, which is a read mapper, the full pipeline, not only sequence alignment, we gain 10x speed up and not 130x speed up, because based on Amdahl law, if you accelerate one part of the entire pipeline, then you expect the speed up to be lower. Again, everything is freely available. You can access it, you can download it and play with the implementation. So GateKeeper conclusion, there is a significant performance gap between high throughput DNA sequencer and read mapper, as we show. Sequence alignment is computationally expensive, but unfortunately it's unavoidable. You cannot just simply replace it with another algorithm because it's the most correct and optimal algorithm to do the sequence alignment, or to figure out where are the edits and what type of edits optimally. GateKeeper is the first hardware accelerator in that time, 2015, architecture as a pre-alignment filter for quickly rejecting dissimilar sequences. It provides a huge speed up of up to 130x compared to previous state-of-the-art software solutions. So as we can see, FPGA pre-alignment filtering greatly speed up read mapping, but remember, FPGA acceleration alone is not enough if you are not tweaking the algorithm to be FPGA friendly, for example, then the benefit will be greater. And we achieved 10x speed up of state-of-the-art mapper, mapper. The FPGA-based pre-alignment can be integrated with the sequencer such that you do the filtration inside the sequencing machine, and that would reduce the data movement rather than moving the data from the sequencing machine to the storage, to the main memory, and then do the filtration at the CPU side, for example. And I'm going to give you a nice story about this toward the end of the lecture. By doing so, by having the FPGA board where we do the filtering inside the sequencing machine, we can hide the complexity of all these designs. Then end user don't have necessarily to deal with these devices. And in this way, we enable real-time filtering. So as we get the data, we are filtering them inside the FPGA, and then we give the user only the correct data. This is more about the shifted Hamming distance, another paper from our group, so you can download it if you're interested in the CPU code and SIMD implementation. You can play with it as well. Let me check. I need to connect the laptop. Okay, sorry. Okay, so that was the paper, and this is the source code. And now this is done with the presenting the paper. It's a bit more than the time, but it's fine. Hopefully we can keep each presentation from the student side within 1 hour or maximum 50 minutes. And now starting with the analysis or your own contribution, hopefully it will be in 20 minutes. It's okay to exceed the time if the discussion is interesting, hopefully. So the strength we find in this paper is this is a new and simple solution. You can see it's based on only bitwise operation to a critical problem in genome analysis, which can affect even medical application and clinical setup. So we have presented new algorithm and hardware architecture. GateKeeper does not sacrifice any of the aligner capabilities because we do not change dynamic programming algorithm. We do not affect it. So we only reduce the workload, the input workload to dynamic programming algorithm. And the design is scalable. So you can see that you can fit more processing cores in the FPGA if you have enough FPGA resource, such as lookup table, or if the DRAM bandwidth can provide more data to the FPGA to process. And some sequencers use FPGA, especially Illumina. So they have already FPGA boards inside the sequencing machine to do some process with base calling and such. So GateKeeper can be integrated inside these FPGA boards. Hopefully the author understands and highlight the limitation of GateKeeper this is a nice thing about all our papers. We normally show the benefits, the pros and cons of our own proposed method. We are not trying to hide the disadvantages of any of our presented work. We greatly improves filtering speed and accuracy compared to all existing work. And this work, basically, when we started, it was the first hardware acceleration to pre-alignment filtering. And after that, we start to see a lot of papers on the same topic. So it's really spurred quite a few papers that build on top of GateKeeper. That is not important strength, but anyway, it's well written, interesting, and easy to understand paper. It's not the most important. So normally we sort this list of strength based on the importance. For the weaknesses, remember, don't try to avoid rat holes. So we are not going to discuss the workload, for example. The benefits of such mechanism require an FPGA and advanced knowledge with computers. This may be problematic for some users that do not have knowledge about the FPGA. But when we are looking about the great benefits, about the application itself, then these issues are not really important. It's okay to spend more time to hire more people to deal with these complex things, but at the end we get huge benefits, especially for the medical field. The amendment of the random zeros, which was highlighted by Daniel, is a simple hack to reduce the number of false positives. But there is no explanation why GateKeeper only flipped the pattern 101 and 1001. What about the other patterns? Again, it's heuristic, but a deeper analysis can be done on this issue. The paper can be confusing at times due to the use of supplementary material. So almost in every section, we refer to the reader to go to the supplementary material and check further details. But at the end, we are limited by the number of papers that we can produce or publish in this journal. But for the supplementary material, normally it's unlimited, so you can attach whatever material you feel the reader might be interested in reading them. GateKeeper accuracy, that is something I didn't tell you about, decreases exponentially for E above 2% and becomes ineffective after E above 8. So when E equal 9, for example, then the filter is not going to filter anything, basically. Why is that? Because we use AND operation, and when we generate a lot of masks, you will have zeros almost everywhere. Then when you do the AND, you almost cancel all the ones you have anywhere, then you are not going to filter anything and you are going to accept these sequences. But since in our paper, even the title, we are targeting short reads, and I told you in practice, we use 5%. E equal to 5% of the sequence length, then we will never use something above 5. So we will not reach 9%, for example. However, in our next work, we improve this and we enable to have unlimited value of E. So in the next few works that I'm going to show, hopefully we have time for that, which is a weakness here I'm mentioning, that GateKeeper is tested using short reads. That was not a problem in 2014 because we have no long reads at all. ONT start to produce their reads or their machine in 2016. And that time we have bad accuracy. The accuracy was up to 60%. So 40% of the read, you are not sure if it is correct, if it is coming from genetic variation or sequencing errors. So we were not targeting this technology back in 2014. The thought and ideas. So this is another paper from our group where we explain all these bottlenecks and we try to analyze the existing algorithm that try to tackle each and every different problem in accelerating read mapping. So I would also suggest it as a reading. Going back to the bottleneck that I mentioned where we move the data, we try to reduce the data movement between different components of the computer. So we have some work try to replace the CPU algorithm with sophisticated hardware, specialized hardware such as FPGA GPUs. We have a bunch of work on that. GateKeeper was the first one on the list. And then we try also to do near memory and in-memory pre-alignment filtering. And we have 3 papers on that. And we also try to accelerate sequence alignment. So for those who were asking about doing the alignment on the FPGA, we did that with the specialized ASIC or near memory processing. So to extend the work, which I was presenting here, several paper. So can we improve the filtering accuracy? I was telling you that GateKeeper cannot work for E higher than 8. Then in this work, we call it magnet. And we try to not amend and not doing the AND operation at all, but rather directly count the number of matches accurately. So if you remember the green highlighted segments out of the mask, rather than doing AND operation to them, you can go through the mask, each mask individually, and try to count the largest number of zeros. Whenever you have those, you save them somewhere, you cancel the surrounding area of that mask, and you move next to the next segment and so on. And that was presented in this example. If you still remember this, the mask we generated, this is exactly the same as GateKeeper, but we changed the way we handle these, the trailing and the starting and the end of the mask. We have some reasons and analysis why we do this way rather than calculating them. But when we do the calculation or the AND mask, we don't do AND, but rather try to find the longest segment of zeros, which is highlighted here. This is the longest among all masks. And then when we find it, we AND all the masks or we replace them by ones, such that next time we iterate over the mask, we will not find this region at all. So we totally cancel this region from the calculation. This is no longer here. So you can convert everything to ones. Or you can just skip the indices such that you will not visit this area again. And the next iteration, we try to find what is the second longest segment of zeros. And we find it to be here, which is number 2 to the left side. And we also count the number of zeros over there, store it somewhere, and then cancel the area to the left side. However, if you notice, I'm highlighting both the right and the left together. Why is that? Because once we find the things in the middle, now the two sub-problems, the one here, let me highlight it. The one to the right side over here, oops. Yeah. The one to the right side over here and over there, it's sub-problem, and it's totally independent of each other. So you can solve the one to the right side separately, the one in the left side separately, try to find the longest segment of zero and calculate the total length of all these sub-problems, and then you can decide on the number of matches or mismatches. So this is how we do it for the right side. For the left side, we calculate the total number of zeros, and then we find the ones accurately, because we go to these zeros and basically ourselves count how many zeros in these long segments. We do not try to do the AND operation or any other operation in comparison with the other masks. And that improved the accuracy a lot. I didn't show the results here, but you can refer to the paper to see the accuracy improvement comparison with GateKeeper. So now, how about the scalability? So you can think about that here. We are handling the entire mask. So we visit the entire mask and we try to count the length of the segments of zeros. But how about if we want to scale it to a sequence of length, for example, 2 million or 3 million? Then we need to really visit the entire mask, try to find the longest segments of zeros, and then the next mask, and so on. So we try to improve the scalability in the next work. We call it Shuji, which is the Japanese door where you slide it. So in Japan, the doors, you need to slide them to open them or close them. And this is similar to our algorithm where we need to slide over all masks. So we need to move over a subset of the mask, calculate the things, move to the next subset, and so on. But we do this independently of each other. And we fix the length of the window we are visiting the masks. So let's see this example. Hopefully it's more clear. So now how we calculate the diagonal, assumed by some equation that is already present in the paper. But the main idea here is how we calculate the segments of zeros, which are highlighted in yellow here. So the correct segments are in yellow. So we want to find 4 zeros over there and single zero over there. That should give you the total number of matches, which is 9. And the sequence length is 12. 12 minus 9, 3. Then you have 3 matches, which are basically between the yellow highlighted segments. So we have 1 here, 1 over there, and 1 over there. That are 3 mismatches. Let's see if Shuji can detect these 3 matches correctly or not. 3 mismatches correctly. So the first one, we have a window, search window of size 4. So you can see in each diagonal is in length of 4. And we iterate over all masks. And we go in diagonal shape. We count how many zeros exist in each diagonal. Not consecutive, but how many zeros. So we count here 0, 1, 1, 1, 4 zeros, 1 0, 2 zeros, 1 0. And we pick greedily the largest number of zeros. So we find that the main diagonal in the middle, the one has the largest number of zeros. So we pick these zeros and we stored them in what we call Shuji bit vector. Then we move next to the next window. We do the same thing. We count how many zeros on each diagonal. We find that there is one of the diagonals has 3 zeros, and then we move them to the bit vector. We keep doing this same, but remember, since these windows are overlapping, there are at least 3 bits overlapping in every 2 windows. So we greedily again store the one provide the largest number of zeros. So before we have 4 zeros here where we store them. And now the next iteration give you 3 zeros and 1, which are coming from this diagonal. Now the 3 zeros here that are overlapping, we replace them only if the new segment that we are going to store will provide larger number of zeros. So think if the previous iteration was 1 here, then in the new iteration we got 3 zeros, then we store the 3 zeros and we replace this one with 0. Why is that? Because we choose to underestimate the number of ones rather than overestimating. Remember, underestimating is always okay because it will produce false positive. So if the errors are 4 and we said they are 3, then our filter going to accept it. And then the dynamic programming going to verify that or double check what decision we have made. But if the number of errors were 4 and we said 5, that exceeding the threshold, then we are incorrectly rejecting that pair. So our decision was wrong. So just to be on safe side, we're always going to underestimate. But remember, this algorithm is very accurate. So the underestimation here or underestimation rate is really low. And we verify this comprehensively with large number of data. So this is just to give you an ideas about the algorithm that you can do and implement in FPGA and where computer architecture can greatly speed up the important problems. But if you propose efficient algorithm that is hardware friendly. So more details in the paper. Now we still have the FPGA CPU communication bottleneck. So it makes sense to use something like processing in memory, processing near storage, processing while sequencing, and so on. All these compute paradigm may alleviate the data movement bottleneck, but not solving it fully because you still need to move some data, especially if you're going to dynamic programming where it's implemented for CPUs and so on. So in Grim filter, we did a totally new filter that is implemented for processing in memory. So we try to do the filtration inside the DRAM or inside the 3D stacked memories. This is more technical details about implementing the paper. If you're interested, I recommend everyone to check the paper. But this is the cube where we have what we call 3D stacked memories. These are DRAM layers stacked on top of each other on the same package die. And here we have the logic layer where we implement the computation that we are targeting. And we are accessing the data vertically from each DRAM bank or within the vault. So our goal is to implement the filter inside the logic layer where we can handle the data from different DRAM layer in parallel and quickly. So there are a lot of constraints about the logic layer. You cannot fit whatever you want because there's energy limitation, there is size limitation for the logic layer and so on. So it's a bit of challenge to design a filter over there. I'm not sure if we have time to go over the filter itself. So I would really recommend you to watch my other lecture from computer architecture class. I'm going to give you a pointer towards the end of the slides to get more ideas about each filter. I explain every single filter that presented today in more details in that lecture because it was 3 hours just focusing on the filtering algorithms. But yeah, let's skip this slide for now. But it was really nice to implement these filters inside the memory where you don't need to move the data further to the CPU and do the filtering there, or we don't need to move it to another FPGA board where you do the filtering over there. But we have a little problem in that design where the reference genome cannot fit inside the 3D stacked memory due to the limited storage over there. So we have high bandwidth communication, but the storage is limited. So for that, we cannot access the locations from the reference genome inside the memory. So we still need an algorithm over the CPU to tell you where are the location that you would like to compare with. So the subsequences generated from the reference genome were generated at the CPU side. Then we communicate them to the 3D stacked memory, and then we do the comparison over there to do the filtering. So that was limiting the performance of such filter, and we solve it later on in the next work. So that leads to faster, more accurate, more scalable pre-alignment filtering, which we call it Sneaky Snake. This is one of our recent work that we try to address several problems that we face in multiple acceleration for pre-alignment filter. And this time, this was the first filter implemented for CPUs, GPUs, and FPGAs. And everything is publicly available, open source, even for the GPU implementation and the CPU as well. So feel free to check it out. And the idea was, the correct alignment is a sequence of non-overlapping long matches. These non-overlapping long matches segment are exactly the things were highlighted in green, if you remember in our GateKeeper example. So these segments were highlighted in green are part of the correct alignment, part of the correct matches between the two sequences. And we find a similar problem in VLSI chip design, where you would like to do single net routing. So to solve single net routing problem in VLSI chip is very similar to what we have in pre-alignment filtering. How is that possible? So this is one example of VLSI chip. If you dissect the chip, you will see a lot of components within the chip, and there is a lot of connection between these modules. And the single net routing problem is as simple as this. Imagine you have an input pin over this side, and you want to pass through it, or you want to connect a path where you, for example, you want to connect a VDD or ground or a new component or whatever the purpose was. And you want to detour through the entire chip to connect the input pin through some component all the way to the output pin. So you want to pass basically from anywhere here all the way to this side. However, you have some restrictions. You want to pass through the minimum component. You want to find a path that you have no obstacles. So each of these components, we call them obstacle. So you want, whenever you face an obstacle, you detour, another obstacle, detour, keep going straight until you reach this side. Each obstacle we consider as edit. So the number of obstacles that you could face through this optimal path is your total number of edits. And based on the threshold, you can decide if your found path is optimal or not. Or basically in another way, if the sequence pair are similar or dissimilar. This is a very interesting problem when we solve it. And when we match it to VLSI design, we find an interesting algorithm to solve it, and which we present in the paper. So first, the bit vectors we generated here are totally different from what we have in GateKeeper or Shuji. We have an equation to calculate that. So I think the equation is a bit clear how you calculate it. You can see it's independent of any other cell. So you don't refer to any Z over here. You never visit any cell in the dynamic programming again. So if all these are Q, which is the query, and this is R, the reference, but you never visit any other cell. You don't see any Z or any visiting to the table Z or the matrix Z, which confirm that there is no data dependencies at all. Whenever you calculate any cell, you don't need any information except the character from the query, character from the reference, and your edit distance threshold. And I have a nice cartoonish example to show you how it works. Let me try to find my cursor first. Okay, it's back. Okay, assume we already know what's the equation used for building this matrix, and our edit distance threshold is 3. Now switch every 1 into an obstacle and every 0 in available path. So every 1 into an obstacle, black box, every 0 into available path with white box. So you'll get this at the end, a lot of black boxes and white boxes. And now you want to pass from the entrance all the way to the exit. You have the snake want to pass. So what you do basically, you iterate over the rows. You count how many consecutive steps that you can move forward, exactly as we do in VLSI chip. You move in a straight line because it's always the shortest path. And whenever you find an obstacle, you detour to another straight line. However, because you want to pass through minimum number of obstacles, you always want to go as far as possible, because as far as possible allows you to pass through minimum number of obstacles. So that's why we pick this path rather than the other alternative path, and we are now here. Now, since we face an obstacle, we reduce the number of threshold now to 2. It's no longer 3. So in the previous slide was 3, now it's 2 because we already face an obstacle. So now it's allowed to face 2 more obstacles. This is the most we can afford. Now we keep finding another straight line, and we find this path has 4 more steps, and we face an obstacle at the end. And this was the largest path or the longest path that we can pass through. So we pick that one, we reduce the number of obstacles, now it's 1, so we can afford only one more obstacle. Otherwise, these sequences are dissimilar. We repeat, we find an alternative path over there, and we pick that one. Now, in this case, we find multiple paths with the same penalty, so all of them have 1 step to move on. It doesn't matter which one to pick, because we always pick straight line. Now, in reality, what we calculate, not this entire matrix, but rather this matrix. Why is that? Because here, since there's an obstacle in the beginning, I don't know if you can see my cursor again, but this obstacle, for example, does not allow me to pass through in straight line. So no need to calculate all these things. So once you face an obstacle in the beginning, don't continue calculating. That's why we have in the next slide, whenever there's an obstacle in the beginning, don't calculate the rest of the row. This makes it very efficient even for CPU implementation, because the number of cells to be computed very less compared to all existing filters. Now we got great performance from this. Even with FPGA implementation, you can see the resource utilization are very low. And this is the accuracy comparison between the other filters. You can see the scalability, or how it scales with edit distance threshold. This is the x-axis, the edit distance threshold. You can see even with edit distance threshold of 10, the sneaky snake is the blue, and the red, and the black. So with different configurations. So the black, for example, providing the highest accuracy. It's over here, even with edit distance threshold 10. And I can tell you, based on experiment shown in the paper, it can scale to 50, for example, or 2000 if the sequence length is really high. So it can really scale well with higher edit distance threshold without causing any problem with the filtering accuracy. All right, that is the result we achieve. So takeaways, a novel method to accelerate sequence alignment in genome analysis. which is using sequence alignment filtering or pre-alignment filtering. And let me delete the annotation. Simple and effective. You can see most of the operation are really simple. And with the help of hardware, software, co-design, cooperation, we achieve a huge speed up. And that is a good potential for work building on it to extend it. So you can see you may come up with new ideas to improve all these filters or the way you implement them or different hardware platforms. That was easy to read and understand, hopefully. And I think by this, we came to the end of GateKeeper or any paper you would like to present. This is how you should present it. You have background, you have the method, the evaluation, the weaknesses, the strength, and towards the end, you need to prepare discussion starters. So you need to have questions for the audience to discuss with them. And since the time already passes, I'm going to continue for about 15 minutes or so. Yeah, thought on the previous ideas. Do we have any ideas to improve on top of the previous filters? So just to help you, rethinking alignment and pre-alignment. So now we separate the pre-alignment filter step and the alignment step from each other. Now, what if we find a way to integrate the pre-alignment with the alignment?
Sorry, I have a quick question.
Yes, go ahead.
In the summary of the GateKeeper, you mentioned that that the E, when E gets bigger than 8%, that its effectiveness plummets. Did you test out different lengths of zeros to substitute with ones to, if you increase the number of zeros to still be accurate enough, or?
Yeah, that will be interesting, basically, to check, but unfortunately, we didn't test that, especially when we proposed new filters. So we were not thinking to improve GateKeeper because we proposed new algorithm to do much better than GateKeeper. But that is something to look at, especially that GateKeeper is based on AND operation, which is really quick, very fast to operate compared to Sneaky Snake, for example, or Shuji.
Okay, thank you.
But that's a good direction, good point to improve the accuracy. You can see it here from this figure at 9 and 10. This is GateKeeper, it's me highlighted. You can see it accept everything. It's over there. These 2 points for GateKeeper, it really accept everything. So it's it's not effective. Shuji is somewhere in the middle. But again, in practice, we are using somewhere here. So even GateKeeper is still good. That is for sequence length 100. But if we move to 1,000 or million, then the 5% would result in, I don't know, 50, 500. The e-value of 5% will be 500. Then when we go to that much of E, no longer GateKeeper or probably Shuji will not work. But for Sneaky Snake, we tested already for a million long sequences and it operates really well. All right, that was a nice idea to improve GateKeeper. So back to the discussion point. Yeah, think about alignment and pre-alignment filter. Until now, we are handling the filtering really independent from the alignment. So whatever we calculate in the pre-alignment filter, we never reuse it in the alignment step. We never reuse this information in dynamic programming. So think about these green highlighted segments. They already mean that there are exact matches somewhere between the sequences. So why not exploit this piece of information to do the dynamic programming part or to do the sequence alignment? So you don't really need to calculate again dynamic programming within these green highlighted segments because they are already accurate where you detect the similarity between two sequences. So you could design something that is alignment aware filtering, for example. That is something to look at if you're interested in improving these. How about extending the solution to longer reads or higher descent threshold? Your colleague just suggested a solution for that. You could look at different ways of doing the AND, for example, or different ways, different patterns to amend these zeros into ones. Or you could try to investigate if you chunk the read into multiple subsequences. If you divide the read into subreads and then handle each separately, such that you see the relative errors within that subread, and then calculate the total number of reads. Probably this is something to look at if you want to improve GateKeeper, for example. Is this solution clearly advantageous in some cases for GateKeeper? So do you see the advantages of using filtering all the time? So when to not use filter, when to use filter, that is also something to investigate. So you need to examine the data before you decide what the distance threshold to use, what kind of filtering to use, and even to use filter or not. That is something also to investigate. Data movement, again, is still a bottleneck. How could we try to reduce it? So in GrimFilter, we try to reduce that by doing some filtering inside memory. But remember that the reference genome was divided and moved to the filter. So the data movement is still not totally solved. But there are some ideas to reduce the data. First, placing the accelerator closer to the memory. Using newer and faster I/O, for example, faster than PCI Express, or could be alternatives to that. Closely integrate the accelerator into the sequencer, as you can do not only filtering inside the sequencer, but also the dynamic programming, such that you really reduce the data movement efficiently. You could offer cloud computing. That's not going to reduce the data movement, but within the cloud, you could improve the data movement by having an FPGA board over there to do the processing. So you could do that efficiently, but you still need to move the data from the sequencer to the cloud, do the processing, then send the result back. And we were thinking that this is very effective, but when it comes to the first few months of COVID-19 pandemic, we see that how the internet cloud is overwhelmed with data movement. And most of the time, the cloud was down for a lot of companies because of several requests to access it. And we cannot handle that efficiently. All right, so now a new work from our group on how to improve Sneaky Snake to alleviate data movement bottleneck. I'm not sure if Gagan is still with us. Probably can go over this quickly.
Hi, Mohammed. Yeah.
Hi, Gagan.
Thanks, Mohammed. Hi, everyone. So I'm Gagan. I'm a postdoc at Safari. So as Mohammed mentioned, this is one of the latest work we did on near memory computing using like real systems with the FPGAs. This work was recently published in IEEE Micro, where we tried to accelerate pre-alignment filtering by taking leverage of FPGAs which have the high bandwidth memory. Mohammed, can you go to the next slide, please? So yeah, so as Mohammed discussed before, the problem is that read mapping is like heavily bottlenecked by data movement from the main memory. So when the memory is far away, then you always have to move data from the memory, and this off-chip data movement is one of the biggest contributors to the power consumption. So the goal was in this work was to somehow reduce this data movement. So what we did, we tried to leverage this state-of-the-art FPGA accelerators with HBM memory. And what we did, we tried to do the processing close to where data resides. So in that sense, you don't have to go off chip, or you don't have to like waste energy and power consumption in moving the data. So what we did, we carefully redesigned the accelerator logic for this Sneaky Snake. Yeah, my video is now clear. There's a lot of like light from outside. Yeah, so, and what we did, like we leveraged the high bandwidth memory, which allowed us to have a logic which was on the FPGA side much closer to the memory. And also what we did, we leveraged there's a lot of heterogeneity in the memories as well. You know, on the FPGAs, the state-of-the-art FPGAs, you can have a lot of different memories. There's VRAM, there's BRAM, You can use LUTs and like flip-flops, everything. So we tried to exploit that. We tried to make a heterogeneous memory hierarchy, which was tailored to the data access patterns of Sneaky Snake. And also we took proper care of like accelerating in terms of like using proper pipelining, partitioning the data, and having enough parallelism. So that's how we took advantage of it. And then we saw like tremendous speed ups. Maybe you can go to the next slide, please. Yeah, right. So here we have like on the left, it's the runtime performance and also the energy efficiency. So what we did, we scaled the number of processing elements, which is how many engines we can fit. I don't know why it's this. I guess now it's okay, right?
Yes.
Yeah, cool. Yeah, so what happens with these, it's not that straightforward with this HBM memory that you just place the accelerator close to the memory, but also they have like new interfaces. So what they do is like conventionally, we have like 1 or 2 channels, which are like for the DRAM, DDR4 kind of memory. But with these HBM, you have really massive amount of channels, which offer a really high bandwidth as well. So we need to think about a way how you will exploit those number of channels and how would you design your accelerator so it scales perfectly. So what we did, on the x-axis, you can see number of processing elements, which is like how many like engines, how many like different Sneaky Snake accelerators we had, and we scale with the number of HBM channels. And then we saw like we compared it to IBM Power 9 system, which is like one of the state-of-the-art HPC CPU systems, which is used in like a lot of supercomputers as well. We compared the performance like when you run Sneaky Snake there, and on the other hand, if you offload it to an FPGA, which is close to HBM, and we made a comparison, we saw like tremendous gains in terms of performance and energy efficiency. So yeah, this is like one of the ways you can take advantage of near memory computing on an actual system as well.
That was great. Thanks, Gagan.
Thanks,
Do
Mohammed.
you have questions for Gagan? Yeah, we are near the end of the slides. So this is another work from our group, very recent one. We call it Gasm, where we do the acceleration, not only for filtering, not only for sequence alignment, but rather for multiple steps together within the same design. So we did an efficient design, and our goal was to accelerate approximate string matching, which is the things I told you about in the beginning. That is a fundamental step in almost all genomic applications. So it's almost to find an application in genomics where you don't compare genomic sequences together. And by enabling acceleration of sequence alignment itself, you could use the same algorithm for accelerating many more applications other than read mapping only. And our goal was to design a fast and flexible framework which can accelerate multiple steps of genome sequence analysis. And we have 3 key ideas, basically. The first one is to exploit the high memory bandwidth and the logic layer of 3D stacked memory to perform highly parallel approximate string matching in the DRAM chip itself. And the second idea was to modify and extend an old algorithm. It was proposed in 1992, so almost more than 30 years back. It's really nice to see how you can enable very old algorithm where the authors didn't think about the usefulness of this algorithm in that time. But in this time, because of the technology change, we find that this algorithm is very effective for this kind of hardware. So you can think about, even if this old algorithm, it might still provide benefits. However, the algorithm was a bit limited, especially with supporting long reads and with supporting traceback and with being parallelizable. And we solved these issues in this work in Gasm, and we improve upon PyTab algorithm. The third idea was to co-design our modified scalable and memory efficient algorithm with low power area efficient hardware accelerator by processing the data where it resides without moving the data to the CPU, for example. And the use cases for Gasm were first read alignment, which is the step I was explaining throughout the entire lecture, and the pre-alignment filtering as well, edit distance calculation, and many other steps that can be used with the same algorithm. So we discussed many of these use cases in the paper in details. The key results first, we tackle or evaluate these three applications. For read alignment, you can see the speed up is really high, even compared with hardware accelerated ones. So I'm not sure if you know about Darwin and GenX, which are hardware accelerated read mapping. And we did the comparison in terms of energy and throughput, and same with CPU-based read mapper, such as Minimap2, which is state-of-the-art, and BWA map. For pre-alignment filter, we compare with Shuji, and we see that the speed up is a bit significant, but the FPGA was really capable, the algorithm was simple, then it was making sense to implement it on FPGA, especially if we solve the CPU FPGA data movement problem with Shuji. I think the Shuji will be almost same performance as Gasm. However, when it comes to edit distance calculation, the speed up is really huge compared to idlib and even hardware accelerated design called ASAP. And you can think about other applications that can be enabled by the same design. So yeah, now let's go back to the discussion starter. I think this is the last point we will discuss here. Can you think of field that could be similarly in need of string alignment as in read mapping in bioinformatics? So it could be any other research field where we need to compare sequences, not necessarily genomic sequences, but you can see that you can classify these sequences into similar and dissimilar, then probably you need a filter for that. Then you may use the same algorithm we propose for other research field. So think about natural language processing or error correction and many more. This is one work, for example, where you combine filtering with dynamic programming as a pre-alignment filter, but with knapsack problem. So it's very interesting to see how you can accelerate almost all dynamic programming algorithm by using some heuristics or some kind of filtering. Okay, the last point for now, it's the adoption of hardware accelerator. So we present a lot of hardware acceleration work, but how about the adoption? So this is the review that we got from one of the reviewers when we published GateKeeper, when we submitted to Bioinformatics Journal. One of the reviewer, and by the way, we got 6 reviewers, which was the first. I'm not aware of any paper got this much of number of reviewers. I don't know why, but anyway. Yeah, for the review we got from the guy who was saying there has been little to no adoption at all of previous specialized hardware solution related to improving the speed of alignment. His mindset was great because he was saying also, although this is a major concern, but it's not a problem with the manuscript at all. And he was discussing why people are not adopting hardware accelerated. And most of the algorithm used in this field was basically implemented for CPU. So I also included our response to them, and you can read it in detail, but what I want to highlight here is that we said it always takes time to adopt a new or different hardware technology since it requires investment into the hardware infrastructure. And this was true. We realized it actually in the companies that they are doing sequencing 3 years later after we propose our work. So you can read the response in details, but remember from our first lecture, dream and anything will come true. So dream about the application that can be enabled by the hardware architecture, and this application will be enabled later on. Dream about the hardware that you are going to propose, and don't worry about whether people will use it or not. If it makes sense, if it solves the problem efficiently, then why not? Change is always good if it's leading to great benefits. So Illumina, for example, so our work was done in 2015 and published and submitted to the journal in 2016. But in 2018, this company started to use FPGA inside the sequencing machine to do analysis, to do alignment, to do filtering, to do all these things. So when we submitted our work, people were not believing in hardware acceleration work. But these days, even more companies such as NVIDIA start to have GPUs just specialized to do filtering, alignment inside their devices. And that was in 2020, where they acquired a startup from Michigan University called Paraprex to do the filtering and analysis and GPUs. So I expect moving forward to have all future devices where we have processing and memory, for example, in this sequencing machine, or could be some other architecture that we are not aware of. So computing is still bottlenecked by data movement, even with these GPU FPGA accelerated sequencing machines. We still face a data movement problem. So we need to find the optimal place to these solutions. And basically, what are the challenges to adopt new solutions is first to accelerate the entire read mapping because of Amdahl law. If you accelerate only filtering or sequence alignment, then the benefit will be less. You need to reduce the high amount of data movement. So you could investigate how you compress the data, operate on compressed data without the need for decompression, for example. And you need to filter out unlikely reused data at very first component of the compute system. And that's what makes sense. So targeting the storage for filtering might be a very good solution. Develop flexible hardware architecture. The challenge for FPGA is you need to hard code most of the parameters. And what bioinformaticians like is to play with the parameters and discover new application. So that is a challenge, and there is no one unique solution to solve that, but it's something doable. The challenge also for the adoption is that adapt existing genomic data formats for hardware accelerators. So I was explaining about the FASTA format and the FASTQ formats, but these all data formats were proposed really long time ago. And these days, if you want to adopt hardware accelerators, you need to enable more efficient formats that has, for example, bit encoded data and with less information that are not needed for hardware acceleration. You need to maintain the accuracy, that is a must. Providing this is a very fast design, that is not enough. Speed is not always the important thing, but also the accuracy, especially when you are dealing with medical data. There is always cost to pay for hardware acceleration and the long development life cycle. That was for the adoption. What else can be done? Think about the puzzle I was explaining. When you have a puzzle and you want to solve this problem, what if we have multiple puzzles? Because until now, we have only single reference genome for the entire world. So you are comparing yourself with a single reference genome. And this reference genome might not be representative to all population, especially African, where they have more DNA than other people, like at least 10% more genetic material than others, or Europeans, and so on. So you need basically a reference genome that represent a certain population, and then you can compare with that. And we, in Airlift, another paper from our group, we try to tackle this problem. When you already do read mapping for a certain reference genome, and now I give you a new reference genome, and I ask you to repeat the step. So it might not be efficient to repeat the entire thing, especially that the new reference genome has some similarity with the old reference genome. And this is what we are trying to tackle, is to find these differences and similarities between the reference genome and then update the mapping results without the need to repeat everything. Now we still have the open question where to place the sequencing machine, these data, whether to process them, at what component, and so on. Remember, speed up and accuracy is not the only important thing. There are a lot of aspects just are there because the nature of the data. So when you are dealing with genomic data, there are privacy issues, there are accuracy issues, and there are scalability issues where you are target, for example, to do genome analysis for the entire Swiss population to figure out COVID-19 or other disease, for example. So it's still very challenging. It's a lot of open questions, how and where to enable fast, accurate, cheap, privacy-preserving, exabyte scale analysis of genomic data. And remember, one important thing, that most speed up comes from parallelism enabled by both novel architectures and algorithms. This is more material to study if you are interested in this topic. And if you are willing to do research, feel free to reach any of us, and we can assign papers, we can do research together, for example, and so on. There's a lot of materials, a lot of videos to watch if you're interested in this topic. So the slides are already public, available on the course page, so you can access all these materials. And that was all for GateKeeper and many other research papers. Remember, our goal for today was not just to present a paper, but rather give you a lot of insights about this research topic. But when you present a paper, probably will be within 50 minutes to 1 hour at most. Thank you so much for your patience. I'm so sorry for taking more time, but I hope it was for your benefits and your growth. If you have no questions, see you next week. Thank you so much. There's something in the chat. I'm not sure if I missed it, but are we already able to pick the preferences of the paper we want to present? Yes, yes. We will assign the list. We almost finalizing the list today. So I think soon you will hear from us with the paper. I think now we have around 18 students. So we have a bit more time for presentation. So I think we will continue in the next 2 weeks with the example presentation with other paper. And then probably we will start after 3 weeks, the first student presentation, or after 2 weeks, basically. We will start the first student presentation. We will give you more details over Moodle and email. All right. Take care. Thank you so much.